Alright, we'll look at the process of post-transcriptional modifications, which only happens in the eukaryotes. Okay, so the last lesson you have learned about transcription, right? So in the eukaryote, the product of transcription is known as a pre-mRNA, which needs to undergo a series of modifications to form the mature mRNA that will be used for translation. Okay, so if we can see from the diagram here that there is the removal of the introns and there's the joining of the exons together. So this is called splicing. There's the addition of a cap at the 5' end, that's 5' capping, and there's the addition of a poly A tail at the 3' end, and that is the 3' polyadenylation. Right, so all this don't happen in the prokaryotes. Right? So prokaryotes, the mRNA is already functional for translation, right after transcription. But for a eukaryote, the mRNA cannot be translated yet. Okay, so if the pre-mRNA is translated, the protein would be non-functional. Okay, so there must be these 3 processing or modifications to form the mature mRNA. Okay, so as you can see from this diagram, right? So imagine this is the nucleus, right, in a eukaryotic cell, right? So transcription occurs. After that, you have the post-transcriptional modification, also known as RNA processing, to form the mature mRNA. Only then can the mature mRNA leave the nucleus into the cytoplasm for translation by the ribosomes. And so we look at the process of 5' capping first. Right, so you have the 5' end of the pre-mRNA, 5' end, right, where the phosphates are. This is the cap that will be added. It's actually quite a normal nucleotide, right? But then at the nitrogenous base of guanosine, there's an addition of a methyl group, right? That's why it's called a methyl guanosine, right? 7-methyl guanosine, right? That's the identity of the cap. Right, so capping will occur catalyzed by this enzyme, mRNA guanyl transferase. Right, so the cap is added in such a way, right, that the bond between the cap and the 5' end of the pre-mRNA is known as a 5' to 5' triphosphate bridge. Right, so it's a 5' end of the cap with the 5' end of the pre-mRNA. There's 3 phosphates, so 5' to 5' triphosphate bridge. All right, and the function of the cap is to protect the mRNA, right, from enzymes that would digest the mRNA in the cytoplasm. So mRNA does not remains forever, right, in the cytoplasm. Okay, so there will be enzymes to digest the mRNA in the cytoplasm, right? So one of the enzymes is called the 5' exonuclease, which starts to digest the mRNA from the 5' end. So the cap would block this enzyme and slows down the digestion by this enzyme. Okay, and in the export of the mature mRNA, right, to leave the nucleus through the nuclear pores, and this process is facilitated by the presence of a 5' cap, and the 5' cap also helps to bring the mRNA to the ribosomes in the cytoplasm. That is where the binding to the ribosomes can occur. Okay? After this, we will go to 3' polyadenylation first. So I'm skipping splicing first. Okay, don't mind, you just skip to the part of the notes for polyadenylation because the process is quite short. So I combine into the same video. All right. So at the other end, right? So just now we look at the capping on the 5' end of the pre-mRNA. This happens at the other end, the 3' end. So we call that at the termination stage, right, of transcription. After the terminated sequence has been transcribed, there is a polyadenylation signal that would recruit some proteins to cut the pre-mRNA, right? So this cut pre-mRNA, right, would leave, and the polyadenylation signal, right, goes on to recruit or goes on to allow the binding of other proteins known as polyadenylation factors. These factors would then recruit the enzyme poly-A polymerase, which would catalyze the synthesis of the poly-A tail, which is a series, a very long chain of adenosine for nucleotides lah. That's why it's poly-A, right? So this long chain of A's would be found on the 3' end. So this is the 3' poly-A tail, right? So the enzyme poly-A polymerase just catalyze the elongation with only the A's, right, at the 3' end of the pre-mRNA. Okay. The function of the 3' poly-A tail is a bit similar to the 5' cap, where it also protects the mRNA from digestion, but in this case by another enzyme known as the 3' exonuclease, which works on the 3' end. Right, so it starts to digest the mRNA in the cytoplasm from the 3' end. Right, so the presence of the poly-A tail would be a buffer, right? So the 3' exonuclease would start to cut the poly-A tail first. So that leaves the genetic information stored in the mRNA sequence to be intact for translation, right? The export, the leaving of the mature mRNA, right, through the nuclear pores into the cytoplasm is also facilitated by the presence of the 3' poly-A tail. Okay? And as I mentioned, right, the 3' poly-A tail serves as the buffer, right? So the digestion by 3' exonuclease would digest the poly-A tail first. So you can imagine, right, the longer the tail, right, then the longer time it needs, right, for the 3' exonuclease to finish digesting the poly-A tail to go on to digest the mRNA sequence itself. Right, so the longer the poly-A tail, the more stable the mRNA, the longer it remains in the cytoplasm. So therefore, the more translation. Okay, so that's the third bullet there. Right, okay, so that would be for 5' capping and 3' polyadenylation.